September 13th, 1999. The University of Pennsylvania, Philadelphia. 18-year-old Jesse Gelsinger suffered from ornithine transcarbamylase deficiency, a rare metabolic disorder that prevented his liver from properly breaking down ammonia. While his condition was managed through a strict diet of 32 pills a day, he volunteered for a Phase 1 clinical trial. The study was not designed to cure him, but to test the safety of a new gene therapy delivery mechanism for sicker infants. Scientists loaded a corrective gene into a weakened version of adenovirus, the common cold virus. At 10:30 a.m., doctors injected 38 trillion viral particles directly into Gelsinger's hepatic artery. The massive viral load was meant to target his liver cells. Instead, it triggered a catastrophic, systemic immune response that the researchers had never anticipated. Within hours, Gelsinger's temperature spiked to 104.5 degrees Fahrenheit. His blood oxygen levels plummeted as his immune system launched an all-out war against the trillions of foreign vectors. By the next morning, his skin turned yellow from jaundice, and his organs began failing one by one. On September 17th, just 4 days after the injection, he was pronounced brain dead and removed from life support. The ensuing investigation revealed severe institutional negligence. The lead researcher held private equity in the company funding the trial, creating a direct conflict of interest. Furthermore, the university had failed to report that 2 previous patients had suffered serious toxic side effects, and they hid the fact that several monkeys had died during preclinical testing. In the aftermath, the FDA immediately halted all gene therapy trials at the university, and the case fundamentally reshaped federal regulations regarding informed consent and financial disclosures in human experimentation. May 4th, 2001. Johns Hopkins University, Baltimore, Maryland. Ellen Roche was a healthy 24-year-old laboratory technician working at the university's Asthma Research Center. Eager to assist her colleagues, she volunteered for a federally funded study investigating how healthy lungs protect themselves during an asthma attack. The protocol required her to inhale hexamethonium, a medication historically used for high blood pressure that was being repurposed to induce temporary lung airway constriction. The researchers administered the chemical via a nebulizer. What they did not know was that hexamethonium was not FDA approved for inhalation, and the chemical structure lacked standardized safety data for human lungs. Roche inhaled the vaporized compound, expecting a brief, controlled chest tightness. Within 24 hours, she developed a persistent, hacking cough. Her lung tissue was actively disintegrating. The chemical had triggered an aggressive inflammatory cascade, destroying the microscopic air sacs responsible for oxygen exchange. By the time she was admitted to the intensive care unit, her lungs were scarred and stiffening. Doctors placed her on a heart-lung bypass machine, but the tissue damage was irreversible. On June 2, 2001, Roche died of multiple organ failure. The institutional failure was absolute. A subsequent investigation found that the university's Institutional Review Board had approved the study using outdated literature from the 1950s. The researchers had ignored published warnings that hexamethonium caused lung toxicity when inhaled. Following Roche's death, the federal government briefly suspended all human subject research grants at Johns Hopkins, forcing global universities to drastically tighten their internal safety screening procedures. May 8, 1951. The University of South Dakota Medical School, Vermillion. 26-year-old Ardys Pearson, a university secretary, and 30-year-old Jack Clifford, a medical technician, volunteered for an experimental study on sedatives. The trial sought to analyze how new cortisone-based treatments interacted with heavy pain medications. Both volunteers were paid exactly 60 cents an hour for their participation. Dr. Louis F. Michalik, a resident physician just 2 months into his medical residency, prepared the syringes. The research protocol called for an injection of meperidine, commonly known as Demerol. Michalik walked into the laboratory, picked up a multi-dose vial from the storage cabinet, and drew the fluid. He failed to cross-check the label on the bottle. He had not drawn Demerol. Instead, Michalik injected both volunteers with 100 milligrams of methadone, a profoundly potent synthetic opioid. This dose was a massive 10 times the standard therapeutic limit. Within minutes of the intravenous injection, both Pearson and Clifford lost consciousness as the massive chemical overload flooded their central nervous systems. Their respiratory drives collapsed entirely. Both volunteers stopped breathing, while the young resident panicked, realizing his fatal error too late. Pearson and Clifford died on the laboratory tables from acute narcotic poisoning. The university was struck by immediate public outrage, leading to the resident being charged with manslaughter. The disaster forced U.S. medical schools to mandate strict dual physician verification systems for all investigational drug preparations. January 7, 2016. The Biotrial Clinical Research Center, Rennes, France. Guillaume Molinet was a healthy 49-year-old man who joined a Phase 1 clinical trial for a new drug codenamed BIA 10-2474. Developed by the Portuguese pharmaceutical company Bial, the compound was designed to inhibit fatty acid amide hydrolase, targeting anxiety and neurological disorders. Molinet was part of the multi-dose escalation group, meant to test the highest tolerated safety limits of the chemical. For 5 consecutive days, Molinet took a 50 mg dose of the experimental pill. On the 6th day, he complained of a sudden, blinding headache. The staff dismissed the symptom as a minor side effect and did not immediately stop the dosing schedule for the remaining volunteers. Within 12 hours, Molinet's vision blurred and he began losing motor control. The experimental compound was causing catastrophic neurological destruction. The drug had bound to unintended enzymes in the brain, triggering massive tissue necrosis and deep cerebral hemorrhages. Molinet collapsed into a coma and was rushed to the university hospital. On January 17th, doctors declared him brain dead, while 5 other volunteers in his cohort were hospitalized with permanent neurological damage. The investigation exposed a deeply flawed protocol. Preclinical data showed that several dogs had died from brain lesions during testing at similar doses, yet the company pushed forward to human trials without adjusting safety margins. Furthermore, the clinic waited several days to notify health authorities after Molinet first exhibited severe symptoms. The French government restructured its trial authorization laws, mandating immediate global stops across all cohorts the moment a single volunteer experiences an unexplained neurological symptom. May 9, 1952, Seattle University, Washington. 19-year-old Stanley Leedom was an active student-athlete who volunteered for a specialized project managed by the university's biology department. The research aimed to test advanced methods of preserving human blood for long-term storage, a critical priority during the height of the Korean War. Volunteers received transfusions of blood that had been processed using experimental chemical stabilizing techniques. The research technicians prepared a unit of blood that had been held in storage for more than 21 days. During the processing phase, the sterilization equipment failed, allowing ambient bacteria to penetrate the storage container. Unaware of the microfractures in the glass seal, the team hooked Leedom up to the intravenous drip line. The contaminated blood entered his circulatory system at a rapid rate. Within an hour of the transfusion, Leedom's temperature spiked to 105 degrees as his body went into hyperacute endotoxic shock. The specific strain of bacteria, which had multiplied in the cold storage environment, released massive amounts of lethal toxins directly into his bloodstream. Leedom's blood pressure bottomed out, and his heart failed 3 days later. The institutional failure lay in lax laboratory biosafety controls. The university had allowed student researchers to process human biological products without direct oversight from certified hematologists. In the weeks following Leedom's death, Seattle University completely disbanded its independent blood research unit. The tragedy led to the implementation of strict federal laws requiring continuous temperature monitoring and automated bacterial culturing for all stored human blood products.
May 24, 2004, the University of Minnesota, Minneapolis. 28-year-old Dan Markingson was suffering from an acute psychotic episode and was placed under an involuntary psychiatric hold. While confined, he was approached by university researchers to enroll in the CAFE study, a clinical trial funded by AstraZeneca. The trial compared the efficacy of 3 atypical antipsychotic drugs, including Seroquel. Markingson's mother repeatedly warned the university that her son was too mentally fragmented to provide lawful informed consent. She wrote desperate letters stating that his mental state was rapidly deteriorating under the trial's medication regime. The university clinicians ignored her warnings, as the department received millions of dollars in corporate funding tied directly to active trial enrollment metrics. The institutional coercion proved fatal. On May 8, Markingson's mother left a final urgent voicemail stating that her son was on the verge of a violent psychotic break. No one returned her call. Multi-milligram doses of the experimental drug regimen had failed to stabilize his violent delusions. On May 24, Markingson committed suicide in a university bathroom, using a box cutter to inflict fatal wounds. The subsequent state investigation uncovered systemic institutional corruption. The university's Department of Psychiatry had a massive financial incentive to keep patients enrolled, actively bypassing standard ethical safeguards for vulnerable populations. The scandal resulted in the University of Minnesota completely suspending all psychiatric clinical trials for over a year. The state legislature passed Dan's Law, a landmark piece of legislation explicitly banning researchers from enrolling patients in clinical trials if they are under involuntary commitment orders. July 11, 2016, the Fred Hutchinson Cancer Research Center, Seattle, Washington. A 42-year-old cancer patient, enrolled under strict anonymity protocols, joined the Phase 2 Rocket clinical trial. Conducted by Juno Therapeutics, the trial utilized a cutting-edge immunotherapy treatment known as JCAR015. The process involved genetically engineering the patient's own T cells to aggressively target and destroy leukemia cells. Before receiving the modified cells, the patient underwent a heavy preconditioning chemotherapy regimen. The protocol combined cyclophosphamide with fludarabine to clear the patient's existing immune system. Researchers altered this chemical formula midway through the trial to accelerate the treatment timeline. They failed to realize how the altered chemical baseline would interact with the genetically modified T cells. The reaction was immediate and uncontrollable. Once the engineered cells were reinfused, they triggered a massive explosive release of inflammatory proteins called a cytokine storm. The sudden vascular permeability caused fluid to rapidly breach the blood-brain barrier. The patient's brain swelled uncontrollably, cutting off vital blood flow, resulting in fatal cerebral edema within 48 hours. This was the fifth death in the exact same trial. The company had initially paused the trial after the first 2 fatalities, but they convinced the FDA to let them resume after just 5 days, blaming the chemotherapy cocktail rather than the cellular product itself. Following this 5th death, the Rocket trial was permanently cancelled, and Juno Therapeutics completely abandoned the development of JCAR015. The disaster forced the FDA to enforce mandatory, month-long independent safety board reviews whenever a single instance of cerebral edema occurs in cellular therapy trials. August 24, 1901, Las Animas Military Hospital, Havana, Cuba. Clara Maass was a 25-year-old American Army contract nurse who had served honorably during the Spanish-American War. Fascinated by yellow fever research, she volunteered for a series of human experiments led by Major Walter Reed. The study sought to definitively prove that the deadly disease was transmitted exclusively by mosquitoes, rather than by contaminated clothing or filth. The experimental setup was brutally simple. Maass allowed herself to be bitten by laboratory-bred Aedes aegypti mosquitoes that had previously fed on dying yellow fever patients. She survived a mild form of the disease after her first bite in March, leading researchers to believe she was developing a controlled immunity. Eager to confirm their theory, the doctors ordered her to be bitten again by an even more virulent strain. The second infection overwhelmed her system completely. On August 14, Maass developed a high fever, severe abdominal pain, and constant vomiting. Her liver quickly collapsed under the viral load, turning her skin deeply jaundiced. She suffered severe internal hemorrhaging, vomiting black, digested blood as her kidneys failed entirely. Maass died in agony on August 24, 1901. Her death sparked intense public outrage in the United States, as critics condemned the practice of using healthy human subjects for lethal viral experiments. The institutional backlash was immediate, and the U.S. military permanently banned all further yellow fever experiments on human volunteers in Cuba. Clara Maass became the first American nurse to have a federally funded monument erected in her honor, and her tragedy laid the earliest groundwork for international laws requiring documented risk-benefit analysis before human subjects can be exposed to live pathogens. If you enjoyed this video, subscribe for similar ones and feel free to drop suggestions for the next video in the comments.